In this session, we will look at the various technologies that are used to actually map, explore, and understand the brain. So how do we know what we know about the brain? When Phineas Gage suffered his life-changing injury in 1848, we knew next to nothing about brain structure or brain chemistry. Even when H.M. had his fateful surgery in 1953, the surgeon didn't know that the hippocampus was associated with memory. In the past 60 years, biomedical understanding of the brain has progressed rapidly, thanks to case studies like these and the introduction of new technologies that let us map the brain and see the brain at work. We no longer have to wait for autopsies to look at individuals' brains. The key technologies we will look at are the EEG, the PET scan, the MRI and functional MRI, the CT or CAT scan, and TMS or transcranial magnetic stimulation. The oldest technology, which is still used extensively in brain research, is the EEG or electroencephalogram, which was first used in 1924. The EEG is a recording of the electrical waves that sweep across the brain's surface. This electrical activity is measured by placing mini electrodes on the scalp. An EEG is not a map of the brain's structure. In the picture to the right, this young girl with epilepsy is having an EEG to see what happens to her brain waves when she has a seizure. The researcher is looking for any abnormalities. In one of my nieces, an EEG detected that she was having micro-seizures many times an hour, which was affecting her development. EEGs are used in many types of psych research. For example, researchers might use an EEG to compare brain activities of a typical sleeper with an insomniac. EEGs are also used to study how brain activity differs in people with autism and dyslexia. The next technology, the PET scan, or positron emission tomography, was first used in 1961. In a PET scan, the subject ingests temporarily radioactive glucose, or sugar. Active neurons consume a lot of glucose, so hot spots in the brain light up, indicating what parts of the brain are at work. Researchers will typically give a subject the radioactive glucose, then ask them to perform specific tasks, such as calculating a math problem or recalling their 16th birthday. This allows the researcher to see which parts of the brain are most involved in this task. One group of researchers has used PET scans to determine whether hallucinated sounds and real sounds activate the same parts of the brain. In addition, as you can see from the PET scan pictured here, these scans can be used to compare brain activity in people with diseases or injury to normal subjects. The PET scan does not create a structural image of the brain. We will see later that PET scan technology is paired with MRI technology to create functional MRIs. The next technology, the CT or CAT scan, was introduced in 1974. CAT stands for Computerized Axial Tomography. The CAT scan provided the earliest detailed anatomic images of the brain. CAT scans use X-ray technology to identify areas of the brain for brain damage or abnormality. One drawback of the CAT scan is that it works better on bone than soft tissue, and the brain is primarily soft tissue. However, CAT scans can identify areas of swelling and bleeding, and they are much more affordable than MRIs. So they are typically done when a patient comes to the emergency room with a traumatic brain injury. CT scans have been used to compare the brains of alcoholics with a control group, and alcoholics were found to have larger ventricles. The first full-body MRI, or magnetic resonance imaging, was done in 1977. Since then, the MRI and functional MRI have become the gold standard for brain research. Some of you have probably had MRIs after sports injuries. In a brain MRI, the subject's head is placed in a strong magnetic field which aligns the spinning atoms of brain molecules. A radioactive pulse is used to temporarily disorient the atoms, and when they return to normal, they send signals that provide an extremely detailed structural image of the brain's soft tissue. If you've ever seen an MRI of your knee or your wrist, you know that it is a huge series of thin slide snapshots that basically provide a 3D image. MRIs can be used to identify structural brain differences, for example, in individuals who are schizophrenic or who have brain tumors. The functional MRI, or fMRI, takes this technology and combines it with PET scan technology to identify changes in blood flow linked to neural activity. This allows researchers to learn all sorts of interesting things about our brains, like which regions are activated when we lie. Functional MRI technology is being used to study questions like, how do people make decisions? Why is it so hard to quit smoking? How does meditation alter your brain? The use of functional MRI has allowed psychologists to become much more objective and scientific in their approach to studying these questions. One of the newest technologies, introduced in 1985, is transcranial magnetic stimulation, or TMS. With TMS, a large electromagnetic coil is placed against the subject's scalp. This magnet creates electric currents that stimulate nerve cells. The pulse easily passes through the skull, but it only reaches about 2 inches into the brain, so scientists can target a specific area. TMS has been used to measure the connection between the motor cortex and specific muscles to evaluate damage from diseases. Like the EEG, TMS does not create a structural image of the brain. TMS is also used in treatment for various disorders. In 2008, the FDA approved TMS as a treatment for depression. It can be used to stimulate nerve cells in the part of the brain involved in mood control and has shown some success. More research will be done to determine its promise as a long-term cure.